is Minton, and this is Hannah England, and today we are going to talk about the potential of CRISPR technology in the field of dental medicine.
Let's dive right in. As of 2025, common dental practices included dental fillings, veneers, and dentures as a way to resolve dental loss and tooth decay. As far as prevention strategies go, there are none that are 100% effective. People can maintain a healthy, low-sugar diet, brush and floss twice a day, and return to the dentist for annual visits. However, there is no guarantee that they won't be affected by some sort of oral disease at any point in their lifetime.
As the science behind genetics advances, we can further improve different parts of medicine, including oral and dental health. Today, we are going to talk about how CRISPR, a gene editing technology, can change the dental industry.
First, let's talk about what CRISPR means. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. This specialized portion of DNA consists of three key characteristics that are important to take note of: the presence of nucleotide repeats, spacers, and PAM sequences.
These components are vital to the function of CRISPR, as they each play an active role in the gene editing process. First, the nucleotide repeats and spacers form the genetic framework that allows CRISPR to identify and target specific DNA sequences, mimicking how bacteria recognize viral DNA.
Now that we have talked about the components of gene editing, let's dive into how it works. During gene editing, the Cas9 enzyme, guided by RNA molecules, cRNA and trace RNA, derived from these spacer sequences, locates the matching DNA in a target organism and makes a precise double-stranded cut at sites marked by PAM sequences, allowing scientists to add, remove, or alter genetic material with high accuracy.
While this explains what CRISPR and Cas9 do, it doesn't necessarily explain how we can use it as a gene editing tool. So, let's look at that next.
In order to use CRISPR and Cas9 as gene editing tools, repair mechanisms must kick in to fuse the two ends together. These two ends are fixed by inserting a sequence of nucleotides in the gap. These sequences can be any nucleotide sequence of a scientist's choosing. In this case, we will be looking at a sequence that would make dental stem cells, dental pulp stem cells, also known as DPSCs, produce more of a beneficial protein, BDNF, or brain-derived neurotrophic factor.
Now, in order for scientific developments such as CRISPR to go into effect, repeated trials of experiments must be run and deemed successful. Therefore, an experiment using CRISPR was performed by the University of Illinois Chicago, New York, University Langone Medical Center, and the University of California, San Francisco in July 2025. The experiment was run on mice aiming to generate dentin, which is the hard, dense tissue that forms the main structure of the tooth. This lies beneath the enamel, which is the outer layer of the tooth, and above the pulp.
Scientists first collected dental pulp stem cells, DSPCs, from human teeth. Using the CRISPR gene editing, they changed these stem cells to produce more of that helpful protein called BDNF, which supports the growth, survival, and function. They then confirmed this change in the lab.
Next, they drilled a small hole into the tooth of the mouse to create a tooth injury. This simulated tooth injury or tooth decay that is often seen in humans. The edited stem cells were then placed into the area that was injured along with the supportive materials. The tooth was sealed to protect it, and after some weeks, the researchers checked to see how well the tooth had healed.
The researchers soon found that the treated teeth grew back with more healthy dentin. This means that the insertion of CRISPR-edited dental stem cells could potentially replace traditional dental fillings.
So, instead of using synthetic filling materials to fill cavities or repair damaged teeth, the edited stem cells are transplanted into the injured area to encourage the tooth to heal itself by growing new dentin naturally. This biological approach stimulates the body's own repair mechanisms, promoting regeneration of the tooth structure rather than just filling the hole with an artificial material.
These applications leverage CRISPR technology's ability to precisely edit genes in dental stem cells or oral bacteria to promote tissue regeneration, fight infection, and correct genetic causes of dental diseases. This broad potential could transform dental treatments by enabling personalized biological repair and prevention strategies.
While there are many positives that come with CRISPR, there are also many downsides. There is also lots of controversy surrounding gene editing technology.
For example, one prominent ethical drawback of gene editing within the field of dentistry is as follows. CRISPR-based dental treatments are expensive and would likely be inaccessible to many, widening health disparities in dental care, especially in underserved populations.
Additionally, CRISPR-based dental treatments pose the risk that gene editing will damage the genome unintentionally by cutting or altering DNA in the wrong places. This error is more commonly known as genome vandalism and can lead to unintended mutations, cell malfunction, or death, and even the chance of tumor formation.
CRISPR, as a gene editing technology, can help improve dental regenerative medicine in so many ways. CRISPR allows very accurate editing of specific genes, and it is ultimately faster and more flexible than older techniques. In our case, these older techniques would be the natural DPSCs, dental pulp stem cells.
CRISPR also holds potential in correcting genetic mutations that cause inherited diseases. In dental medicine, this could mean correcting genetic defects causing enamel disorders.
In all, as CRISPR continues to help us develop gene editing techniques, we can regenerate smiles using the power of science. Thank you.
Thank you.